Good afternoon. Here we're going to begin chapter 1 of cellular biology. Hopefully this is going to be familiar to most of you, kind of a review. So let's begin on what is a cell and what makes a cell a cell. Cells are specialized through what we call differentiation. All cells come from stem cells, and then they will begin to differentiate and become a particular cell, like a bone cell or a muscle cell or a blood cell. All of our cells have some major functions, and I've listed these 8 functions here, and we're going to talk about each one of them and what they do to the cell and what their purpose is. So the first one we have here is movement, and movement basically is going to generate any kind of force to cause that cell to move. For instance, muscle cells will contract and get smaller or relax and get longer, and helps with the process of the movement of limbs. Also, when we have muscles that are attached to certain organs, particularly hollow organs, they will allow those muscles to contract and relax to help with the emptying of those muscles, like the urinary bladder or the stomach. Conductivity is the second feature here, and this is where we have electrical stimulation that comes along the surface of the cell. If you remember from anatomy and physiology, we talked about something called action potentials. These action potentials are going to be what is that conductivity. We see these mostly in our nerve cells. Molecular absorption is a function of cells, and they take up nutrients from the environment, from the things that we eat, and they then will utilize those through the process of metabolism. Secretion is going to be a process by where you have a cell that synthesizes particular materials that it takes in from the environment. It's going to convert those to some chemical substance and release them to the surrounding environment. When we have things like waste, this is going to be released from the cell through a process of excretion. So we have a number of cells that are part of our immune system that we call lysosomes, and these are going to break down materials. And when they do, then those substances will be released from the cell, and the waste will be excreted out, and we will release that waste through urination or defecation. Respiration is the process of producing chemical energy from our nutrients. We convert that we convert that nutrient energy to chemical energy in the form of ATP. This is going to take place in the mitochondria. In our cells, it is always going to take place in the mitochondria. In bacterial cells, however, this is not the case because bacteria don't have a mitochondria. But any cell that is not bacterial, the mitochondria is the site of ATP synthesis. Reproduction is the duplication of cells. This is going to allow us to replace old cells, dying cells, cells that have been damaged, and replace them with new cells through the process of mitosis. This is the way that we maintain our tissues, and they're going to take those new cells, replace any old or damaged cells so that we have good viable tissues throughout our life. Communication is a way that cells signal between each other. Our cells have to talk to each other, and when they do that, they send signals through pathways that contribute to the ability of our bodies to maintain homeostasis. So when we have things changing and getting out of homeostasis, we will have communication that will either send us into a negative feedback loop or a positive feedback loop that will take us then back to the state of homeostasis. We can divide our cells into eukaryotic cells and prokaryotic cells. Eukaryotic cells are any cell with a nucleus. Prokaryotic cells are cells without a nucleus, and these are all of our bacteria. You are a eukaryotic cell. Your cell has a nucleus, it has a cell membrane, it has cytoplasm and organelles that have a division of labor. So any eukaryotic cell is going to be broken down into these 3 components. First off, we have the nucleus. This is located generally in the center of the cell. It is the largest of all of our organelles, and its job is to take care of DNA replication. It's the site where we are going to have our DNA housed. It's surrounded by a nuclear envelope with nuclear pores. And this envelope is a double membrane that is continuous then with our rough endoplasmic reticulum. When we make our proteins, we're going to see that our DNA is going to act as a template to make messenger RNA. That messenger RNA is going to leave through the nuclear pores to go to the endoplasmic reticulum. Inside the center of the nucleus, we have a very dense region called the nucleolus. This is where our ribosomal RNA is produced, and we begin the production of our ribosomes that we're going to use in protein synthesis. Again, the primary function of the nucleus is cell division. This is where our DNA is replicated over and over and over again as our cell goes through mitosis and we begin to produce new daughter cells. It's going to also control the genetic information. It's going to determine when is it time to replicate and when is it not. Some of the other functions are to repair the genetic material through the process of replication. When we go through DNA replication, if there are mistakes, there are enzymes within the nucleus that are going to fix any of those mistakes. Transcription is the first part of protein synthesis. This is where we're going to take our DNA, we're going to read one strand of it, use that strand as a template to make our messenger RNA in the first part of protein synthesis. Within the cytoplasm, we also have organelles, and that cytoplasm is kind of that gelatinous aqueous solution within the nucleus and the cell, between the nucleus and the cell membrane. It essentially is going to be nearly half the volume of the cell, lots and lots of fluid in the cell, so that we have a way that our organelles can remain buoyant within the cell itself. The organelles are going to be things like the nucleus, the endoplasmic reticula, the mitochondria, the Golgi body. These are all going to be within the cell, have a particular function within the cell, and they will then do their job and send the product off either to another organelle in a division of labor or send it out of the cell to do a job outside the cell. The cytoplasm is also where we're going to store a number of our macromolecules, particularly our fats and our carbohydrates. We're going to store these in these things called secretory vesicles. These secretory vesicles I like to call your cellular pantry. We store our foodstuffs that we're going to eat for later in our pantries in our kitchen. These secretory vesicles are going to store fat and carbohydrates that we can use later on for energy or for the building of other types of molecules. The plasma membrane is the outermost membrane of our cell. It's selectively permeable, meaning that it gets to decide what goes in and what goes out of the cell. I like to call it the gatekeeper. It's going to determine what molecules come in and what molecules go out. It also defines the shape of our cell. Our cells are going to have a very particular shape, and that plasma membrane helps define that shape. Our plasma membrane is made up of a very peculiar molecule called a phospholipid. That phospholipid is what we call amphipathic, meaning that part of it, its phosphate head is hydrophilic, and its fatty acid insides are going to be hydrophobic. So when we put this together in a phospholipid bilayer, we have a charged inside and outside of our membrane so that it can interact with the watery extracellular fluid and watery intracellular fluid. And then sandwiched in between, we have these fatty acid tails that are going to be hydrophobic, and it keeps fluid from going in and out at random. It also is going to spontaneously organize itself within its inner and outer layers. The phosphate heads are going to face outside to the extracellular and intracellular fluids, and those fatty acid tails are going to be sandwiched in between. It also allows for a barrier to the diffusion of water. That hydrophobic inside is going to keep water from being able to go across that membrane and flooding the cell or causing the cell to rupture. So this is going to serve then for that barrier of water moving across and allowing for the cell then to essentially die. Within the plasma membrane, we also have proteins. These proteins are made up of amino acids, and they are there for structure. Some of them are going to stiffen the membrane, but some of them are going to act as proteins that allow for the movement of fluids and other substances to come into and out of the cell. We can divide these into our integral and our peripheral proteins. Integral proteins are going to be proteins that go from the outside of the cell all the way to the inside of the cell. It goes all the way through the bilayer. It has integrated itself all the way through the bilayer. The peripheral membranes, these are proteins that are not embedded all the way through, but rather on one side or the other. They are on the periphery of the plasma membrane. They usually, however, are going to be bound to an integral protein and act in the function of that integral protein. The function of our plasma membrane is going to be determined in large part by some of these proteins that we have in the membrane. Some of them can act as receptors that will recognize substances and allow them to come in or out of the cell. Some of them are pores, or what we call transport channels. These are going to let things like charged particles, like our electrolytes, that are going to come in and out. We can also have charged amino acids or charged proteins moving in and out through these transport channels. Some of them can act as specific enzymes, and these enzymes will drive certain portions of the organelles within the cell. For instance, our sodium and potassium pump is going to be driven by some of these plasma membrane enzymes. This is going to allow for us to maintain the proper concentration of sodium outside the cell, potassium inside the cell. Some of them are going to act as cell surface markers. A lot of these are made up of particular proteins. Some of them will have lipid or carbohydrate moieties or parts to them. A good number of our surface markers are made up of glycoproteins, and these are the identity proteins that tell our tissues from foreign tissues, or our self from non-self. Our cells also have to join together, and so they have to adhere together. So we have molecules in the cell membrane called cell adhesion molecules, and these are going to allow cells to join together, attach to each other and to the cytoskeleton of the cell, and allow for that plasma membrane then to hold together and communicate with other cells, or join together with other cells. And then finally, some of these membrane proteins will act as a catalyst to activate our chemical reactions that are going to take place within that cell membrane. So our cell receptors are going to be proteins that are on the cell membrane that are going to bind to specific molecules that we call ligands. These ligands are going to join to a specific portion of that receptor and activate that receptor. This ligand has to fit the receptor perfectly, much like puzzle pieces together. If you have a piece of a puzzle that fits in not quite right, it doesn't, the picture doesn't become clear. But when we put all the puzzle pieces together and all of them are in their right place, the picture on that puzzle becomes clear. Same thing with these ligands. They have to fit exactly so that these receptors work the way that they are supposed to. Many of these membrane receptors are going to poke out of the cell membrane. They're going to be on either the outside or the inside of the membrane. And we can classify these receptors by their location or function. A lot of times, the receptors that we have in a particular cell that are active in that particular cell are going to determine the cell's functions. Receptors will also determine the ligands and the response that these have. So for instance, if I have a sodium receptor, that sodium ion is going to bind to that receptor. It's going to cause something to happen in that receptor that's going to change that receptor, which will then activate a function within the cell. So our receptors, our ligands, and the responses are all inextricably linked. Cell adhesions, these are mechanisms that are going to allow our cells to bind together to make tissues. If you remember your structural hierarchy, once we get to the cellular level, then we go to the tissue level. There are three types of cell adhesion or cell-to-cell adhesion molecules, and we have these in the plasma membrane as well as some of the other connective tissues. The three types, we have cell adhesion molecules, we have the extracellular matrix, And we also have some that we call specialized cell junctions. Your book has a number of really good pictures, and I would take some time to go and look at these individually so that you are familiar with the different types of cell adhesion molecules. In the extracellular matrix, we have proteins. These are long, stringy, fibrous proteins that are going to act like glue to hold these together. They can also provide a pathway for nutrients and waste to move through those tissues. If you remember some of your tissues from A&P when you went through the tissue portion, you have areolar tissue. This is a very good example of that extracellular matrix and the pathways that are left within those tissues. The matrix is going to be made up of three different structures, three different macromolecules. We have our fibrous proteins, which can include proteins like elastin and collagen. We had adhesive glycoproteins, which are going to generally be fibronectin. And then we have our proteoglycans and that hyaluronic acid. Collagen is going to give us our strength to our tissues. It's what we call tensile strength, meaning that I can pull on those tissues. I can give it that stress of being pulled on, and it won't tear or it won't break. When we have diseases like osteoarthritis, one of the things that we see is this collagen breaks down, and this is going to contribute to some of the symptomology then of osteoarthritis. Elastin is more of a rubbery type protein. It's like elastic. We can pull it, we can stretch it, and it will go right back to its original structure and recoil. We find this in a lot of our hollow organs that have to stretch, like our lungs, our bladder, our stomach, lots and lots of elastin there. Fibronectin, this is a glycoprotein that anchors the cell. It's going to make sure that the cell stays where it's supposed to be and doesn't migrate off to some other part of the body. In metastatic cancer, what we see is that this fibronectin has let loose, and these cancer cells now can move throughout the body and find themselves somewhere else and now produce cancer in another portion of the body. This is basically what happens when we have metastatic cancer. These specialized junctions are going to be where the cells come together and are connected. These cell junctions have two functions. First one is to hold all the cells together so that we can maintain that tissue. And then some of these cell junctions are going to permit certain molecules to pass through from one cell to the next, so we can communicate one cell to the next. There are three different types of cell junctions. You've got the desmosomes, the tight junctions, and the gap junctions. The desmosomes I liken to the snaps on a shirt or a belt. Desmosomes are going to unite cells together. They're going to join the cells together, and they're going to brace and maintain the cells for stability. If you look at the picture here, you'll see at the top here, we have what we call belt desmosomes, and these are usually found at what we call the apical or top surface of our cells. Then throughout, we have places where we have spot or button desmosomes that will hold those cells together, kind of like the snaps or the buttons on a shirt. Tight junctions are going to be areas within the cell. If you look here, we see we've got this very dark area here. This is considered a tight junction. This tight junction is a barrier to diffusion. Nothing is going to get past here. If you look down here at this picture here, you can see there's nothing going to go past here. Then we have gap junctions, and this is where we have a small gap in our cell membrane where we have these coming together. You can see this gap here and this gap here. This is going to allow for smaller molecules to pass through the membrane, and these can act then as communication channels, and we can then get information from one cell to the next. Cellular communication is essential. If we don't have good cellular communications, we fall out of homeostasis. Homeostasis, when we fall out of homeostasis and we no longer have that stability, then we begin to see disease. So communication is extremely important in that process. Communication is going to happen when we have plasma membrane signals that are going to be sent out and bind to receptors. When we have receptor proteins inside any kind of target cell, they're going to receive information and send more information out. And then we're going to have the formation of protein channels that are going to coordinate any kind of communication or activity between our cells. So all of our neighboring cells know what they need to do, know what their job is, and we don't have any kind of miscommunication that might draw us out of that homeostasis. Any kind of alteration in this communication is going to be the onset of disease and progression. So we have a number of different modes of communication. First, we have contact dependent, and that's exactly what it sounds like. This is where you have close membrane to membrane contact. They have to come together. There's going to be some kind of receptor that's going to receive a signal, and we have that membrane contact. Paracrine signaling is going to secrete some kind of local chemical. We're going to have a target cell that is within the tissue, and the secreting cell is going to be in that same tissue. So this is why we call it a local communication. So even though the cells are not the same, the secreting cell is going to send out its information, and the target cell is going to receive it, and both of those cells are in the same tissue. Autocrine signaling is when you have the cell sending out the signal and receiving that signal in a particular receptor. We see this in our T cells when we activate our immune response. Then we have hormonal signaling, and this is specialized to the endocrine cells. They're going to secrete a hormone. It's going to be sent out into the blood, and it's going to then be taken to the target cell that is a much farther distance away. Think about your pituitary gland in your brain sending a signal to the thyroid gland in your neck. So there is a distance away there. Neurohormonal is going to be hormones that are going to be released by neurons. They're going to also be carried in the bloodstream to a target signal. And then finally, we have the neurotransmitters. These are going to diffuse from the nerve to the cell directly. We have the presynaptic neuron is going to send a chemical, that neurotransmitter, to the postsynaptic neuron or the postsynaptic target cell, and we're going to then have communication then between those two cells. We see this between our nervous system and our muscles, between nerves and nerves. So hopefully you remember this from A&P. Cellular metabolism is the sum total of all of the chemical reactions that are happening within that cell. Some of those are going to be where we're taking in large molecules and breaking them down into smaller ones, like proteins into amino acids. These are catabolic reactions, and they always produce energy. Anabolism is just the opposite. We're taking those smaller molecules and we're building large ones. We're taking our amino acids, we're joining them all together and making a big protein. These are going to require energy. So catabolic reactions produce the energy to make available for anabolic reactions to build larger molecules. The overall process of ATP is essentially taking glucose and oxygen and having those chemical reactions break that glucose down to carbon dioxide and water, and then leaving us with energy in the form of ATP. Catabolic reactions are going to be reactions that are going to break down any kind of foodstuffs that we eat. And this happens in 3 stages. First, we have digestion. This is where we're going to take things into our mouth, we're going to chew them up, and we're going to start breaking them down into smaller subunits. This happens outside the cell. This is going to be activated by secreted enzymes within the cell. Think about when you smell food, what happens? Your mouth starts to water. When you put food in your mouth starts to water. So we have enzymes within that saliva. You have enzymes in your stomach, in your large and small intestines. All of these are going to be activated through this process of digestion. Glycolysis and oxidation, this is going to be a process where we're going to take that glucose that has been liberated from the things that we eat, and we're going to split it down to produce a smaller molecule called pyruvate. That pyruvate then can go in many different directions. It can go into anaerobic respiration, it can go into aerobic respiration, and it can go into fermentation. And it depends on the tissues that this process is occurring in as to which direction this new pathway is going to go. If we go into aerobic and anaerobic respiration, the next pathways are going to be the Krebs cycle and the electron transport chain. The Krebs cycle is going to take that pyruvate and release any potential energy that is in there through the process of oxidation and reduction. This is going to give us two molecules of what I call potential energy in the form of NADH and FADH. Now, this is not energy that we can use. This is kind of like money in a safe deposit box. Unless we have the keys to open them, we still can't use this energy. But we can take it to the electron transport chain where they have the key to open this up. When we go through aerobic respiration, NADH and FADH are going to be converted to ATP through a process called oxidation and reduction, and we're going to get an enormous amount of ATP left. NADH is going to yield anywhere from 2 to 3 ATP, and FADH will always give us, for 1 FADH, we'll get 2 ATP. So we are making a lot of ATP with a little bit of investment of our potential energy. I'm hoping that you remember some of this from A&P. If you have questions about them, please write it down and we'll go over it next time in class. When we look at the process of ATP formation, we are essentially going through what we call phosphorylation, where we're adding a phosphate to ADP. ADP is adenosine diphosphate, and then we're converting that to ATP through adding a third phosphate, so we end up with ATP, or adenosine triphosphate. This phosphorylation mechanism happens two ways, either through what we call substrate phosphorylation, where through an enzyme catalyzed reaction, we release a phosphate from a substrate and we give it to ADP. This requires no oxygen and is going to happen generally in glycolysis and the Krebs cycle. When we have oxidative phosphorylation, this is going to remove a hydrogen from our NADH and FADH through the process of oxidation, and we're going to then use the energy related to that to drive the phosphorylation of ADP to ATP. This is always going to take place in the mitochondria. This requires oxygen because oxygen is what we call our terminal electron acceptor, and we must have that oxygen in order to run this process of oxidative phosphorylation. Membrane transport also has mechanisms by which we can take nutrients in and we can have waste come out of the cell. This is going to be driven by the nutrients that are being moved. So we're going to move these from an area of high concentration to an area of low concentration. Or if we need to, we can go backwards, go upstream, where we're going from an area of low concentration to an area of high concentration. So any of these areas where we're going from an area of high concentration to an area of low concentration, this is what we call passive transport. There's a concentration gradient that moves these molecules from low or from high to low, and it's kind of like rolling a ball downhill. We don't have to move anything. We don't have to help that ball down the hill. It moves down on its own. This is going to allow for the movement of water across the membrane through the processes of osmosis. We can have simple diffusion, where we have nutrients moving across that membrane. No energy is required. That's why we call it passive transport. Active transport is just the opposite. We're going from an area of low concentration to an area of high concentration. This means that we are moving up the concentration gradient. This would be like us being at the bottom of the hill and having to force the ball back up. So it's going to require some kind of energy. We also have processes that we call endocytosis and exocytosis. These are going to also require energy because they're requiring the movement of the cell membrane. So we have a number of different types of passive transport. The first we have is what we call simple diffusion, and this is going to be the movement of a solute from an area of high concentration to an area of low concentration. This concentration gradient is going to allow for this molecule to move across the membrane, and the deeper or steeper the gradient, the faster that molecule is going to move. Filtration is going to be the movement of water and solutes through a membrane due to hydrostatic pressure. Now, diffusion, the difference here is the addition of water. Solutes are going to be the only thing that are diffusing through with simple diffusion. Filtration is going to have the hydrostatic pressure pushing the water against the membrane. And this is basically a mechanical force. Think about some of these water bottles that have membrane filters in them, and you put your water in and you sit and you have to wait a moment before the water starts moving through that filter. And what's happening is you're building up hydrostatic pressure on the other side of that membrane so that there's enough force to push that water through the membrane. Finally, osmosis is the movement of water down a concentration gradient, no solutes this time. In osmosis, the membrane has to be permeable to water, or more permeable to water than it is to other solutes. So there are portions of the membrane that is their whole job, is to push water in and out of the membrane. The concentration of solutes on one side of the membrane is going to drive the movement of that water. And so the greater the solutes on one side is going to essentially pull that water to that side. I want you to write this down. Wherever salt goes, water follows. So wherever my concentration of solutes is higher, water is going to follow to that side. I'm sure you learned about tonicity when you were in A&P, and this is essentially the effect that water has and concentrations of solutes have on the movement of water within a cell. And we look at something called osmolarity, and this is the concentration of molecules per water or per weight of water within a solution. So when those are equal, we say they are isotonic. So when my extracellular fluid and my intracellular fluid have the same concentration of molecules, or my osmolarity is the same, we're going to see that water moves from one side to the other equally. If I have a hypotonic solution where my extracellular fluid has a lower solute concentration than my intracellular fluid, we're going to start pushing water to the inside where the solutes are greatest. When we push water to the inside where solutes are greatest, my cell is going to begin to swell. Finally, if we have a hypertonic solution, we're going to see that my extracellular fluids are higher in concentration than my intracellular fluids. So this is going to force water from the inside of the cell out, and this is going to then cause that cell to shrink up. Both of these can cause changes in the way that the cell is going to react within a state of homeostasis. We can start to begin to see the breakdown of certain things when we have issues with tonicity. We also have membrane transport. This is basically going to be a facilitated diffusion. This is where we have the passive transport, so no energy here, but for some reason the molecules can't cross the membrane. So they need one of those channel proteins within the cell membrane to allow for the movement of this material to go from one side to the other. There are three different types. You have the symport, the antiport, and the uniport. The symport, if you remember from A&P, is a transport protein that will allow two molecules to go across in the same direction. The antiport is going to allow two molecules to go across in opposite directions. And then the uniport is going to take one solute across in one direction, whether it's in or out of the cell. Finally, active transport is the last type of transport, and this is always going to require energy. A good example of this is the sodium-potassium pump. Getting sodium back out, we're going from an area of low concentration inside the cell back to a high concentration outside the cell, and potassium at a low concentration outside the cell back into where we have a higher concentration inside the cell. This allows us to get back to resting membrane potential. Endocytosis and exocytosis are two ways that the cell is going to utilize ATP. The first one, endocytosis, can be either what we call phagocytosis, which is cell eating, or penocytosis, which is cell drinking. Both of these are going to require a change in the cell membrane, and when that happens, they produce pseudopods that's going to allow for that cell membrane to produce these kind of false arms, and that takes energy. Exocytosis is going to be the expulsion of waste out of the cell. You're going to have some kind of secretory vesicle bind to the cell membrane, and that cell membrane now is going to change its shape, open up, so we can now send whatever is inside that vesicle out into the extracellular fluids. Cell reproduction is a process by where we go through and take old cells and replace them with new cells in this process of asexual reproduction called mitosis. Mitosis has two phases. We have the mitotic phase, which is nuclear division, and then cytokinesis, which is cytoplasmic division, where we're going to start dividing out the organelles within the cell. So we have a process here where we're going to see that the cell is going to go through several phases. We go through the S phase, which is the synthesis phase, where DNA is going to be synthesized, and we're going to look at this, and we're going to look and make sure that everything is correct. We're going to make sure that our DNA has been replicated accurately, and it's going to be correct. I'm sorry to say that two of my things are in out of order. So we're going to go from the S phase to the G1 phase. The G1 phase is what we call a gap phase. This is a phase in which the cell will look at the new pieces of DNA, make sure they are good, so that we can go on to the next phase. This is basically a period between the mitotic phase and the start of DNA synthesis. This is going to allow for us to make sure that everything is okay, so we don't send a cell on to go be reproduced that has something wrong with it. I like to call these gap phases our molecular stop sign. After the G1 phase, we're going to go to the mitotic phase. This is where we're going to divide out our genetic material and begin to go into the cytoplasmic division, where we're going to start dividing out the organelles. When we get through the mitosis, then we're going to go into the G2 phase. This G2 phase is where we're going to have RNA and protein synthesis happening within the cell. So we have the appropriate proteins and protein synthesis mechanisms to build up any of the proteins that we need in the cell before cell division is complete. Within the mitotic phase, we have 5 different phases. The first one is interphase, and this is a long phase, and these are going to be different for every cell type. The first interphase, this is where we're going to have the chromatin begin to coil up. It begins to thicken and shorten. This is when our cell is in its active phase. Then we're going to start going through, and the cell is getting older, and we're now going to begin to go through cell division. So a lot of you learned the acronym PMAT for mitosis. So we have this first phase, which is called prophase, where the spindle fibers and the centrioles are going to begin to be active. Those centrioles are going to move to the polar sides of the cell. Spindle fibers are going to start to be forming from those centrioles, and now we're going to see the nuclear membrane around that nucleus begin to disappear. These spindle fibers are going to bind to something called the kinetochores of the chromosomes, and this is going to allow us to pull the sister chromatids apart, the 2 different copies of our DNA. Before we start to pull these apart, though, we're going to go into metaphase, where all the chromosomes are going to line up down the center of the cell, and they're all going to be attached to spindle fibers. Once they're all lined up, those spindle fibers are going to begin to shorten. And when they do, that begins to separate the sister chromatids within the chromosome. And we're going to start to see invagination of the cell membrane, where we're going to be at the very beginnings of cytokinesis. Once anaphase is complete, we have 2 identical sets of chromosomes on opposite sides. We're going to start going through the replication cycles. The spindle fibers are going to start to disappear. We're going to start to rebuild that nuclear membrane, and cytokinesis is going to be complete, where we basically come in and split the 2 daughter cells. Now we have 2 cells that are genetically identical. Within cell reproductions and cytokinesis, we have these things called growth factors. These are signals between the cells. We also call these cytokines, and they are going to regulate the growth of our cells. We need these so we don't have unregulated growth of our tissues. Unregulated growth is never a good thing. These growth factors are going to act to overcome any kind of intracellular braking mechanism. They are going to keep the cell from restraining any kind of cell growth or block any kind of progression into the cell cycle that shouldn't be happening. One of the cancers that has a defect in these types of cytokines is a cancer of the eye in children called retinoblastoma. The retinoblastoma protein is defective, and we don't have that intracellular mechanism that regulates that tissue growth. Tissue formation is going to be the formation of cells that are different, creating a tissue. So we have multiple different types of cells making tissues, and those tissues come together to make organs. To form a tissue, the cell has to have some kind of intracellular recognition and communication with its neighbors so that we can have some adhesion and adherence so that we can make this tissue. We also have to have some memory so that we can remember what cells are within what tissues, and we don't start attacking our own tissues. In A&P, you should have learned that there are 4 different types of tissues. You've got your nerve tissue, epithelial tissue, connective, and muscle tissues. We're going to talk throughout this semester about each of these individual tissues and what happens to these tissues when things go wrong within the cell. Okay, that's the end of this lecture. We will pick up on genetics in the next class. So please make sure as you listen to this lecture that you write down any questions that you have, and we will answer them as quickly as we can.